And it's my pleasure to introduce Clay Rains. He's a fish biologist at the USGS Eastern Ecological Science Center, the National Fish Health Research Laboratory in Kearneysville, West Virginia, and a PhD candidate at West Virginia University. Additionally, he is now the lead of the Functional and Molecular Bioassay Core Technology team as part of the USGS Environmental Health Program. So in addition to fish health and genetics research topics, the Functional and Molecular Bioassay Core Tech team applies and modifies microbial and eukaryotic reporter bioassays to detect the presence of bioactive chemicals in environmental water samples. Looking forward to your talk, Clay.
Awesome. Thank you for the introduction, Nick, and great presentation, Cheyenne. A lot of what you said directly applies to all of this, and important to note that, for the most part, a lot of times we aren't working on model organisms. We're working with wild fish. And so the questions and how we actually work with these fish health questions totally changes. I'm going to talk about a couple of topics directly related to my dissertation research, specifically the identification and detection of novel viruses. And the wild fish comment is a little bit of a misnomer for some of my stuff, but we'll get to that later. And I also wanted to take this time to acknowledge a pile of co-authors. Without them, I wouldn't be half of where I am today. And this is a team sport, and I'm just really glad to be part of such a cool team. So moving on to topic number one, we're going to start with river herring, novel viruses of river herring. River herring, in this case, referring to the anadromous alewife, and blueback herring to anadromous alocene fishes. In general, wild river herring seem to be on the decline, and there's a lot of work being done to investigate the potential causes for this. But in general, fish health is part of the question, as it typically always is. Simultaneous to a lot of this work happening, a parahepatin was discovered in white suckers. And similar evidence seemed to support the evidence of a hepatinovirus within river herring. So the question, of course, became, are we seeing the same effects that we saw in white sucker within these river herring populations? And outside of knowing that it existed, a whole lot of other questions had not really yet been answered. So that was kind of the starting point on a lot of this work. In brief, the methods were essentially as follows. Subsamples of liver, and in some cases, homogenized viscera, were collected for DNA extraction. DNA was extracted and submitted for rolling circle amplification. This enriched the circular genomes, including circular DNA viral genomes. Those enriched genomes were then tagmented and prepped for sequencing using an Illumina platform primarily. Those short reads were then assembled via CLC Genomics Workbench, annotated, and open reading frames predicted in Genius Prime. And that allowed us to create a qPCR assay, specifically a TACMAN assay. We were kind of able to identify potential additional candidates which might be suitable to obtain more genomes. And ultimately, we were successful. We were able to identify multiple draft genomes from alewife specifically, and identify the alewife hepatitis B virus. We actually designed that qPCR assay focusing on core protein, as every other open reading frame actually had either synonymous or non-synonymous SNPs. We sampled from 4 different locations, including a landlocked population of alewife in Lake Hopatcong, and fish from the Delaware River, Great Egg Harbor River, and Maurice River. Pairwise comparison of the P protein of alewife HBV placed them pretty squarely within the metahepatinoviruses, and similar to the Astato-tilapia metahepatinovirus and Mexican tetrahepatinovirus, and also fairly similar to a bunch of Lake Tanganyika cichlid hepatinoviruses, and also kind of illustrated the differences between those fish meta and parahepatinoviruses. 2 different looks here at the qPCR results. 2 major takeaways. 1, there's a pretty good spread on the data. And 2, detections were only among 2 different populations of anadromous alewives, and only migrating adults. Great Egg Harbor River and Maurice River both contributed samples that were qPCR positive, No young of the year were above detection, and none of the late Hopatcong landlocked population. So just a lot of stuff to consider there. If you're interested in reading more about this, the recently published manuscript is available in the most recent copy of viruses. I'm going to move on to topic number 2, which is novel adenoviruses of black basses. Cheyenne kind of mentioned this is one that is a major topic of interest within the Chesapeake Bay watershed, and ultimately has been identified kind of nationwide. If you've ever followed any of Dr. Luke Iwanowicz's presentations or any of my presentations, I'm certain that you've heard me talk about blotchy bass syndrome in some way. Essentially, when this work first started, we knew that there was this condition, this highly visible external lesions that are visible hyperpigmentation. We identified a virus associated with the condition in smallmouth bass. And then the assumption was that a similar virus or the same virus would probably be causing the identical presentation within largemouth bass and other black basses, which have also been reported. But outside of that, that was kind of the objective moving forward. Do we know anything more about these? How are we detecting them? No one's been able to culture these things. How else can we address these questions? And as I kind of mentioned, this is kind of cutting edge. So being on the cutting edge means that you have a lot of stuff kind of developing on the fly, and it's resulted in a lot of spin-off projects that are in various stages of progress. The ones that I'm going to talk about in any level of depth today are ones that are in bold and underlined, but there's also a bunch of other really cool stuff going on that I'm always interested in talking about. If you have any questions, just reach out. But ultimately, major takeaways are we had more than the 2 smallmouth bass adenoviruses that we originally started with on the board. We then quickly obtained a largemouth bass adenovirus that was organizationally very similar to the smallmouth bass adenovirus, but only about 70-ish% identity, so totally different critter. And even though organizationally similar, knew we had to start looking elsewhere. Abbreviated methods, again, fairly similar workflow to what we did with alewife work, except for in this case, we were able to implement a non-lethal sampling protocol due to the fact that these are visible external lesions. So we started using repurposed buccal swabs as dermal swabs, collecting DNA from those, extracting DNA using the same rolling circle amplification, same tagmentation process and sequencing workflow via Illumina. And also the same annotation pipelines. We also used a different bioinformatics software on this, these viruses called C-Note Taker 2, which does a really good job specifically on virus work. From that, we were able to develop yet another qPCR assay, this one a cyber-based assay, and kind of continued to feed the beast on that work. In general, we also had, as a result of this stuff being highly visible, some simultaneous and parallel workflows happening where we actually had multiple citizen science data collection efforts happening. So we ultimately used both smartphone images and a smartphone application to collect kind of spatiotemporal data of where people were seeing fish that exhibited this incredibly characteristic appearance. And then those high quality images were good enough quality that we could then subsequently use them downstream for additional analysis. So I kind of mentioned this stuff just kind of keeps spiraling and getting out of control. So we started with 2 in smallmouth bass. We quickly added MSA1 in largemouth bass. And even then, we added 2 more adenoviruses, MSA2 and MSA3. This is hot off the press, hopefully coming to a publication near you very soon. But ultimately, we know that these groups of adenoviruses are kind of closely related to one another, but outside of that, still just kind of putting them on the board. We were able to obtain an entire draft genome of MSA2, and sharp-eyed observers might notice that the nomenclature and of the annotations has actually changed over time, and that these are more up to date and more in accordance with the protein naming conventions rather than just where they fall out within the cassette. But in general, within MSA3, we did not obtain a full genome yet, but we got enough to begin working on it and starting to look for it to try to get it in higher numbers. But wait, you know, as the stuff goes, there's always just something more popping up. As a result of using this rolling circle amplification method, we're really good at amplifying circular genomes. This includes mitochondrial genomes or any other circular viral genomes kind of along the way. This includes MS-NDV, a newly identified Echidna virus. That will also be kind of paired with this new data set, hopefully coming out soon. And in general, this Echidna virus, for those unfamiliar, Echidna viruses are essentially hepatitis B viruses without an envelope protein. So they are quote unquote naked. And structurally, this Echidna virus is very similar to others already known. And as I mentioned, we were able to get enough of all 3 largemouth bass adenoviruses to design primers and begin testing, just kind of see what was happening. And as well as with that Echidna virus, which we obviously obtained the whole genome of. And thus far, we've only had one unlucky soul out of a couple hundred fish evaluated so far that has actually had a co-occurrence of all 3 Micropterus salmoides adenoviruses, as well as that Echidna virus. Here and there, we'll get pop-ups of 1 or more, but never as many as whatever was happening with this individual, which was non-lethally swabbed and released into the wild. And I kind of mentioned earlier when I was talking about authors, but I would not have been able to do half of what I've done without such great partners. I was very fortunate as a result of an earlier AFS Fish Health section presentation to partner with Bass Pro Shops and Texas Parks and Wildlife. During much of this work started during the pandemic, and Bass Pro Shops actually stepped up to the plate and helped us collect visible samples. Texas Parks and Wildlife really kickstarted a lot of our citizen science work that I'll discuss briefly. As I mentioned with Bass Pro, we actually reached out to 156 different retail locations and ultimately ended up with 32 complete swab kits returned. These 32 complete, meaning that they contained both blotchy and normal swabs from normal skin of clinically hyperpigmented fish. So we were able to kind of run a comparison between normal skin versus this hyperpigmented skin to see what kind of differences popped out. And looking at the qPCR data, there were some differences, at least particularly within MSA1. 2 main takeaways from this data set. 1, if the fish looks splotchy for the most part, there's a lot of reasons for hyperpigmentation, but if it realistically meets that look, you have greater than a 96% chance that you're going to get a positive detection. And also, clinically normal skin, you're still capable of detecting virus, but at lower levels than is visible as illustrated by these violin plots. Detection of MSA2 and MSA3, not quite as dramatic, maybe a little bit of a pattern in MSA3, but not enough samples to really define that relationship as clearly. I mentioned the citizen science effort. Texas collected data for 2 years, essentially having anglers submit when and where they encounter blotchy fish. And a very clear pattern started to arise that basically March, April, May were kind of the hot spots, kind of indicating a prevalence pre-spawn and then a drop-off kind of immediately post-spawn, at least as far as reporting rates. This gave us really good confidence moving forward that we were able to launch the citizen science effort using the smartphone app. At the conclusion of the effort in February of this year, 5,500 black bass were reported, about 10,000 fish in total. And that added another 362 confirmed blotchy bass cases, visually confirmed, giving us an overall prevalence of 6.5%, which is kind of anecdotally similar to what we'd encountered in other data versions. And looking at the reports by month, and just that little small graph there pictured is a reminder, a refresher of that Texas distribution. Essentially the same pattern. We're looking at March, April, May. We also have a little bit more of a pronounced peak in June and July, but keep in mind that that effort spanned the entirety of North America. So we've got a lot longer period of latitude to actually cover there. Coming up towards the tail end here, using these high quality data, we were hoping to train a neural network to be able to identify blotchy lesions and quantify them. We ended up having to do manual annotation, and I was fortunate enough to have 2 AFS Hutton scholars to help accomplish that work. And looking at that work, the pattern is again pretty reminiscent of what we were looking at before. where April, May, and June, you're kind of getting a little bit more higher percentage of blotches, which again probably means there's a threshold at which you kind of just don't notice it as an angler. So that's just kind of a snippet of a lot of the things we have going on. Too many things to mention, particularly in the time frame. Could talk about it for hours. So if you have any questions, concerns, comments, please reach out to me. I'm always available, and thank you for this opportunity.
Excellent. Thanks a lot, Clay. That citizen science aspect of the fish health, the blotchy bass project is very cool, innovative. Folks, perhaps Clay, you had your email there at the end, but maybe you can drop that into the chat. Folks, if you've got a quick question, we'll take it. Otherwise, put them in the chat and Clay can respond there. Okay, thanks a lot, Clay.